Let's start, okay, about the Cyclodextrin experience. Feel free to share in the chat box what you work on, as it's always interesting to see where you're from and how you are attached to cyclodextrins. Of course, as you may know, cyclodextrins have a lot of uses, typically in the field of them being used as excipients for solubilization, stabilization, or taste masking. But today we'll talk about these novel applications that you can see here, especially the ones that are used as active ingredients. Uh, okay. Uh, so, a little warm up as we try to be interactive as much as possible. So, let me know if you know the companies, just very quickly in the chat box, if you know the companies that I show here. So, who knows about Erivin Therapeutics or Renatus, Colrem, Zyversa Therapeutics, or Avaron Pharma? Any of those ring a bell? Because the topic we discussed today is something that's hot and emerging, and these are all companies who not only work with cyclodextrins, but companies who have cyclodextrins as active ingredients among their asset. In reality, what we see is a much larger number. So these on the picture are all companies who use cyclodextrins as active ingredients, not necessarily already marketed, but who have inventions, patents, preclinical, clinical programs, or marketed products where cyclodextrin is the active ingredient itself. Of course, that also involves us. So let's see how this story started. And basically, we'll discuss two, or we will distinguish two main areas because, from the mechanism of action point of view, I'm sorry for my voice, it's some kind of uh, respiratory attack I'm suffering. So, from the mechanism of action point of view, there are two very different approaches how cyclodextrins can be used as active ingredients. So one big group are the scavengers, and here the concept is very similar to the excipient use. So the cyclodextrin is used to encapsulate a compound. The only difference is that what you inject or insert to the body is an empty cyclodextrin, and the cyclodextrin has to find something within the body, capture it within the body, and remove it from the body. That's technically the concept. Of course, in order to achieve it, you need a very specific and a very strong interaction, obviously. So, in that regard, it's different than the excipient use. And the other group are cholesterol modulators, where the mechanism of action is not as clear as with the other one. Here we'll see a very diverse set of diseases where the cyclodextrins are used as active ingredients which have in common some kind of malfunction in the cholesterol homeostasis of the human body, which the cyclodextrins seem to restore somehow. But the how part is still debated and it's not absolutely clear, but I'll get to that point. So how the story started, using cyclodextrins as active ingredients is a surprisingly old story. So as you can see, it's 43-year-old story. This was the first publication by Joseph Pita, who is one of the big pioneers of cyclodextrin research. He was, by the way, the inventor of the hydroxypropyl-β-cyclodextrin, what you certainly know very well. So he was the first one who used cyclodextrins as an active ingredient, and it was the scavenger type of concept. So they used a dimethyl, a methylated cyclodextrin, for the treatment of hypervitaminosis A. So there was a vitamin A accumulation in a kid's body, which didn't have any other treatment, and they injected with this aim, the cyclodextrin. To remove the retinoids, which was sufficient, which was successful, by the way. So it was a success story, but it didn't become a drug in the end. But this was the foundation of the concept. And of course, based on this, Captain Scavenger, as I would like to call it. Bridion was developed much, much later, as you will see. So, uh, here the basic story was that, okay, the first case was an intoxication, but let's assume that we inject something to the body deliberately during a surgical procedure, during anesthesia, we inject a drug, and of course, after the procedure is done, we would like to suspend the effect. And this agent is this neuromuscular blocking agent rocuronium, which is very widely used still in the surgical practice. So we would like to capture all the rocuronium and remove it very quickly from the body. So they started screening different cyclodextrins, which is the good cavity size, and doing all the modifications. So this was a systematic drug discovery type of project, only they were not targeting a receptor, but they were targeting a small molecule with a cyclodextrin, but conceptually, it's the same. So the gamma cyclodextrin was a good fit, but it has to be extended, the cavity, because it was too short for a relatively big small molecule, and they needed some negative charge, some permanent negative charge to capture this emitter group. Um, so that is how Bridion was developed for a very specific binding with the rocuronium. Um, the success of Bridion was not as straightforward as it seemed today because it was a fairly expensive compound compared to the golden standard. So the golden standard was naloxone, which costed like $1 for treatment, and there was Bridion that you could use, which costed, okay, 70 euros per treatment. Uh, but Sugammadex had a lot of advantages compared to naloxone. It acted extremely fast. Within minutes, it could remove all the rocuronium from the system without any clear side effects, very quick recovery, and so on. But it also had some side effects, mainly anaphylactic reactions. And this was the main reason by the FDA, because due to which the FDA rejected first the use of Bridion, and in the EU it was approved since 2008. And only 7 years later was approved in the US, where its success story began, and it became a blockbuster drug eventually. But this is the concept. So as you can see, what they did is that they modified the narrower cavity of the cyclodextrin so that it can bind also with electrostatic forces, the rocuronium, not only via encapsulation. And it was found later that it also works for similar compounds, so it can be used for others. Um, it's now marketed as an MSD product, but in many countries, the original patents already expired. So there are also generic companies on the market. And here you can see, okay, it has a very vivid patent landscape and pretty big sales numbers. So based on the, okay, I'm sorry, I have to mute someone. Sorry. So based on the very same concept that, okay, we can remove something selectively from the body without influencing the intrinsic homeostasis, new developments came, and I'll mainly just list these because the concept is the same. So now you can see that if you have a molecule, you can design reasonably a cyclodextrin to capture that molecule selectively and remove it from the body. That's because of the basic pharmacokinetics of cyclodextrins. If you inject the cyclodextrin, the renal clearance is extremely quick. So we're talking about 1 hour for a complete clearance. So the next disease that is being developed is Stargardt disease. So we see lipofuscin accumulation in the eye. So of course the administration route is a bit different, but conceptually is very same. It was discovered in Cornell University, and then company named Revision took over the development. Then a slightly different concept was developed by Alverum, the company that I mentioned, more precisely first Oakload, then Alverum, who said that okay, we can antagonize the neuromuscular blocking agents, so why not antagonize low molecular weight heparins because they are also their overdose and their presence can also lead to a lot of potential problems. So they also designed a new cyclodextrins for themselves, which they're using as an antidote. The clinical trials, I think, already started, so it's in early phase clinics. Although here I must say that the mechanism of action is undisclosed, so it's not unambiguous that it's a scavenging type of concept. It could be that there is another type of um, antagonizing effect with the cyclodextrin. So that we'll see when there is data uh, available. Then there is another very interesting concept, and still we are talking about systematic drug discovery. So there is the company in California, San Francisco, called Cyclodextrin Therapeutics, uh, who took a very interesting challenge. So what they wanted to remove from the body was not a xenobiotic, so not a low molecular weight heparin or not uh, an anesthetic, but it was an intrinsic molecule. So during the cholesterol homeostasis, an oxidized form of cholesterol is formed, created, so within the body, within all of us, which, as they found, contributes significantly to plaque formation in the so it's related to atherosclerosis and creation of foam cells from macrophages. And their idea was that, okay, we can create a cyclodextrin that's specific for this oxidized cholesterol. So just imagine we're talking about there is plenty of cholesterol within the body, and there is this other version of cholesterol that differs only by one single oxygen from the original molecule. And they said that they can create a cyclodextrin that's selective for this very small difference. And we are not talking about a receptor here, it's a cyclodextrin. Even for a receptor, it would be very challenging. So they visualize this concept that, okay, in order to have that, let's not use one single cyclodextrin, but we combine two cyclodextrins like handcuff that can distinguish then possibly better between cholesterol and 7-ketocholesterol. And as the data showed, it actually can. So there was, I think, a thousand-fold difference between the affinity towards oxidized cholesterol and cholesterol in their targeting. So of course, this is doing something with the cholesterol homeostasis, but it's not cholesterol targeting. It's a sequestering agent, but it's sequestering something that's intrinsic, which is beautiful because for that you need extreme selectivity, which they tackled. So this is also in clinical phase one, close to the end of clinical phase one in Australia, actually. So we'll see how that goes, but the first results are very promising and the rest. So what kind of anything that you can think of, overdosing of drugs and abuse, like fentanyl, other types of intoxications, poisons, of course, I don't know, improper use of chemicals or other type of drugs. So technically, it seems reasonable that you can design cyclodextrins for any specific small molecule because cyclodextrin chemistry is very versatile. There are two cavities that you can modify and so on. And besides these new targets, there are other also companies who developed follow-up molecules for the original target. So for rocuronium, so Adam Gammadex is supposed to do the exact same as Sugammadex, the exact same target. It's just a different compound. So these are the scavengers. Now let's move to the other area, the cholesterol modulators. And this story starts with hydroxypropyl-β-cyclodextrin. So the excipient that you all probably know very well. And of course, this is a well-known oral and parenteral excipient. And what happened is that there was an active ingredient, poorly soluble, that they wanted to use for the treatment of Niemann-Pick C. So what they did is, okay, let's take the cyclodextrin, we solubilize the active ingredient, and we administer to the mice, and we'll see how it goes. And the solubilization went very well. But surprisingly, what they found was when they just tried the vehicle uh, against the mice, the vehicle itself worked exactly as good, or in some cases even better, than the formula that contained the active ingredient. So that's how they discovered that, okay, so the cyclodextrin shall have an effect on its own uh, on cholesterol modulation. However, there were a lot of questions coming out of this study. Apart from it was groundbreaking because in this study, Niemann-Pick C, it's a lysosomal storage disease, and the cholesterol accumulates within the cells. So cyclodextrins cannot penetrate via active transport any biological barrier, only via passive transport, and even then very poorly. So we are talking about single-digit percentage that can go through biological barriers. So it seemed very unlikely that a cyclodextrin can enter the cell or go to the organ, enter the cell, and capture the cholesterol within and remove it from the cell. So it was a very wild mechanism of action to hypothesize, but it seemed to work. And this story went on and on. And okay, we'll see where it is now. So the questions are, is cholesterol really the target here? Because, okay, we are using hydroxypropyl-β-cyclodextrin, which can indeed encapsulate cholesterol. But there are other cyclodextrins that can also encapsulate cholesterol, like the methylated cyclodextrin. As you can see, it can encapsulate cholesterol much better. And it was not working as well as the hydroxypropyl-β-cyclodextrin. But there are other cyclodextrins, like the hydroxypropyl-gamma cyclodextrin, that cannot encapsulate cholesterol at all, or not at all, but to a very small extent. And it works very well with Niemann-Pick C. So are we really solubilizing or encapsulating cholesterol, or we are affecting the cholesterol homeostasis via a complete different mechanism of action? And this triggered a huge debate, of course, still unresolved today. So there are concepts which hypothesize that it's done via autophagy activation. Others claim AMPK enzyme or other enzyme activation, so the cyclodextrin directly acting on certain enzymes. There are a lot of theories. What we know is that hydroxypropyl-β-cyclodextrin works. There are also other cyclodextrins, as we discovered with Kristin Davidson's group, that work very well, and not all the cyclodextrin work. So, and then cholesterol affinity, we also tested not just cholesterol, but other lipids that are affected by the disease. So we couldn't really drive any direct conclusion between encapsulation efficiency towards these accumulated compounds and the treatment outcome. And of course, I mentioned this is one hypothesized mechanism of action, but okay, it raises some questions. How the cyclodextrin goes into the cells efficiently, how it can find the cholesterol, how it can remove the cholesterol from the cells, and so on. So there are a lot of questions here, but still, we have it ongoing. So where is the story now? Because after the discovery, it took uh, a Y-type turn, and there are two directions. So two distinct developments spurred out of this discovery. One was adrabetadex, which is also called VTS 270, which is now developed by Mendos LLC, but okay, it had many owners. So it's a special type of hydroxypropyl-β-cyclodextrin, a purified version of the original compound, and in the patents they claim that it's via autophagy induction, not cholesterol removal. Unfortunately, the program, which was an intrathecal administration of the compound to the CNS. So this cyclodextrin cannot cross the blood-brain barrier, and a lot of the symptoms for the disease were CNS related. So the idea was that why not inject the compound directly to the CNS to alleviate these symptoms? Uh, so the program in clinical phase 2/3 failed to prove meaningful improvements compared to the control. Of course, patients still receive it, and there are discussions that they may be restarted, but this is where it is now. And there is a competitor program or alternative program, which is differs in the most probably in the type of hydroxypropyl-β-cyclodextrin, although here we know little what is exactly used, but we know that it's parenteral administration, not intrathecal, so it's a systemic use of the cyclodextrin. And okay, so I messed up this slide. Obviously, these are not the companies who develop it, but it's called CTD Holding Cyclotherapeutics, who is uh, the holder of the uh, product here. So it's intravenous. They are still in clinical phase 3. They just concluded, I think, the first or the second year program. The results are very promising. Of course, this is a systemic administration, so they see a lot of improvements in the peripheral uh, organs like the liver, lungs, and then less so in the CNS, but it's still promising. As you can see, what I would point out the dose. So can you see per body weight kilogram, 2 gram of cyclodextrin? It's a huge dose. So even if it works, and this was used all in all the animal studies and everywhere, even though it works, it's not very specific, or it has some barriers that the high dose is supposed to overcome because it cannot penetrate the blood-brain barrier, or it's not accumulated in the system. It clears very, very quickly. So this is an excipient, and you should understand that it's an excipient, which means that that okay, it behaves like an excipient, not necessarily as a drug. So it has its drawbacks. And okay, so it was discovered that for Niemann-Pick C it can work and it can affect cholesterol homeostasis. So the question came, what else has issues with cholesterol homeostasis that should be able to influence? So let's try out cyclodextrins there. And cyclodextrins came and they worked in Alzheimer's, clinical phase 1 ongoing, Parkinson's, very solid preclinical data, in familiar hypercholesterolemia, against different type of antidotes. This may not be as straightforward, but here the concept is that the cyclodextrin can disrupt the lipid rafts that are necessary for the work of some poisons, and even FSGS, where the cholesterol accumulates in the kidneys, which was a very sound idea since the cyclodextrin is clearing through the kidneys. So having a high cyclodextrin concentration in the kidney is something that would inherently happen after the administration. So a lot of these ideas and came also the FSGS is, I think it's starting the clinical phase or it has just started recently. So there are a lot of other targets, and this widened and widened and still widens. So there are certain type of cancers where cholesterol is involved. So triple negative breast cancer, colon cancer, also I think pancreatic cancer already have some data showed Kabuki syndrome, which is a rare disease of the brain, gout, lymphedema, even dry eye disease. So there are a lot of applications, all for hydroxypropyl-β-cyclodextrin, so all for the excipient that was available. So the question came, okay, we know the excipient works, but that was more or less the only one we tested. So it's very unlikely that if the cyclodextrin concept works, the excipient is the best choice. The already available excipient cannot be beaten by something that we design. And how does the cholesterol modulation work? So if we don't know how um, it's affecting the cholesterol homeostasis. How do we design something that is needed? So there came a lot of studies where other type of cyclodextrins were evaluated. So the alpha and the gamma versions of the hydroxypropyl cyclodextrins, which also worked in many cases, but companies also experienced or experimented with improving the half-life of the cyclodextrin via polymerizing them or creating rotexanes, which is a necklace of a cyclodextrin, or improving targeting with sugar-appended cyclodextrins so that they can target the brain or certain type of cancer tissues, or peptide conjugated cyclodextrin again to enable them to get into certain organs via active transport and so on. And all these companies and university groups are doing these developments. And then there are other mechanism of actions, which are or other types of cyclodextrins that act a little bit differently. So a few words about those. There is a methyl beta cyclodextrin, which has an application for asthma or COPD, where it is claimed to reverse the surfactant dysfunction. So it's acting on the lipids in the lung, but this cyclodextrin, this methylated cyclodextrin, is at least a very efficient lipid binder, much more efficient than the hydroxypropyl-beta cyclodextrin. It was also found that this methyl cyclodextrin and its derivatives are more efficient in a certain type of cancers. Same mechanism of action, affecting cholesterol homeostasis, but they could trigger cell growth inhibition and apoptosis much more efficiently compared to hydroxypropyl-beta. And a completely new area came during the COVID. Well, originally not during the COVID, the basic discovery was much earlier, but the idea was that sulfated cyclodextrins, and specifically certain sulfated cyclodextrins, can have an antiviral effect. And a new asset was developed by a company called Asterivir from Switzerland, based on this concept, where the cyclodextrins would have a specific heparin sulfate dependent heparinoid type of effect on certain viruses, and it was originally tried for COVID, but later it became a potential asset for influenza and other viruses because it really exhibited a broad spectrum uh, antiviral activity. And as you can see here, we are talking about real serious uh, concentrations that are really drug-like. Uh, So this antiviral application of cyclodextrins has a very broad literature, and basically two type of concepts live there: sulfated cyclodextrins with their heparinoid type of effect, and methyl cyclodextrins for their cholesterol sequestering and virucidal activity. And you can find a lot of papers, as you can see, tested for different types of viruses. I'm not going into uh, huge details, but conceptually they work. They're not super efficient antiviral compounds, but they did work in these cases. And there are other cyclodextrins that were typed. So there was also CRISMAB mentioned that it could be a potential anti-COVID compound along with this study, but later it didn't get to the market. There came an idea that, okay, maybe we don't develop cyclodextrin-based antivirals, but they can help in the prevention, and products actually arrived to the market with that concept. So like mouthwashes, oral sprays, stuff like that. And here the idea is that there is the cyclodextrin B can be a chemical barrier, we could say. So not just use physical barriers when it comes to viruses, but also a chemical barrier. And here there were supporting clinical evidence also for such kind of products. There were two or three of them that were eventually marketed. Um, yeah, and just a few very curious, interesting active ingredient applications. So, cyclodextrins can be, this is called a pore-forming toxin. So, there are toxins that can intersect themselves into the membranes and open ion channels and lead to cell death. And there was a very famous study, and again, this is a very old application of cyclodextrin specifically against anthrax, where the mechanism of action for the cyclodextrin was to block the pore here. So the cyclodextrin was designed for the exact size of the pore so that it would sit into the pore and stay there forever, blocking the pore. And that was a very potent inhibition, by the way, and a lot of nice patents, even companies um, were established based on the concept, but none of them reached the market eventually. You can find a lot of papers on wound healing and cyclodextrin. Specifically, cyclodextrin polymers are very popular for this application. You can create a hydrogel and they can help in the healing process. You can, of course, even incorporate other compounds into the cyclodextrin. So the cyclodextrin themselves also have a wound healing potential, but it can also be combined with others. And there were also companies formed on this concept. And I thought I had one left, but okay, maybe I forgot to add this one. So basically, what we can see here is that summarizing the two distinct concepts. Scavenger cyclodextrins have a very clear target. You know what you want to capture, and for that, you have to design a cyclodextrin. It's a custom cyclodextrin. What you have on the shelf will not work because you want specificity and you want selectivity. And for that, you have to design the cyclodextrin. And that works. Um, Here, and the cyclority is only already leading the way for that. The question is, can we remove something from the body that is not toxic in itself, but toxic at the amount it's present? So there are diseases where, I don't know, we have elevated testosterone levels. So could we use cyclodextrins to remove that to the normal level and not remove all of them? This is something that could lead to a lot of therapeutic options, but still remains an unsolved potential. And there are the cholesterol modulators, where the design is very difficult because we don't understand how they work clearly. Uh, so that's why we usually use repurposed cyclodextrins. Uh, Obviously, because they're cheap and they are available. But still, it's very clear that if we designed tools, so if we understood the mechanism of action and designed something, we could be much more efficient. And interestingly, what I was always curious about, so if we suspect that it's the cyclodextrin that's capturing the cholesterol, why don't we test the cyclodextrins that can capture cholesterol really, really well? That's something almost all the uh, companies out there are negligent to do. Oh, yeah, and this application, one of my favorite, although it's not necessarily a pharma application, but naked cyclodextrin, naked alpha cyclodextrin. So super simple, super cheap. And now we have tremendous clinical data, human clinical data actually, that uh, used on its own as a non-digestible dietary fiber. So this cyclodextrin is on the GRAS list. You can eat it as a food supplement, can improve the sugar and the lipid balance and absorption. So technically what happens is that the alpha cyclodextrin binds the fats from the food after the meal and do not let them absorb or just absorb slower. And there are quite a few products there. So there is a significant literature that this can be used technically as an active ingredient, which enjoys the GLP-1 hype currently. So the other dilemma which we haven't touched, is more of a chemical nature. So when we are talking about cyclodextrins, we have to distinguish between mixtures of cyclodextrins and single isomers. So the cyclodextrins that you use typically during drug formulations are mixtures of cyclodextrins. So Let's put it clearly. So the alpha, beta, and the gamma are distinct molecules. And when, these are single isomers, we could say. They go into that group. But when you create an excipient derivatizing these cyclodextrins, usually you apply a random modification, which means that you don't control the reaction, and the cyclodextrin has over a dozen groups that will react with the agent during these procedures. So the end product is not a distinct molecule, but it's a mixture of hundreds or thousands of molecules. These are what are called random derivatives. The captisol is like this. The hydroxypropyl-β-cyclodextrin is like this. The methylated cyclodextrins are like this. Typically what we use as super excipients. That's why they are so flexible. That's why you can use them to so many compounds. On the other hand, it is possible to control the reaction very much and create derivatized cyclodextrins that are single entities, just like the mother alpha, beta, and gamma cyclodextrins. These are the ones that will be very specific to something, but these are the ones that, okay, are not that easy to make. So this is how you can imagine single cyclodextrins if we see how the methylation is controlled. Here you can see that, okay, we put the methyl groups only in this position, or that, or that. Or we put two methyl groups, but we control the reaction very well. And among the compounds we talked about, Sugammadex is like that, or the follow-ups of the Sugammadex are like that, or the antiviral compound of EPFL is a single isomer like that. So what is in the favor of making single isomer cyclodextrins like this? So you can define very well what you have as an active ingredient. You can define the degradation product of the active ingredient very well. You can define the metabolites of the active ingredient very well. You can develop analytical methods easier. You can do the PK/PD studies easier. You can study the mechanism of action easier with these compounds. Usually as with other active ingredients, the synthesis is more challenging, so we're not talking about a one-step synthesis, but multiple step synthesis and purification and different controls, use of organic solvents, a lot of analytical method development. At the end of the day, of course, these will be more expensive than the random derivatives. So this is what it looks like, a typical chromatogram of Sugammadex. Here you can see Sugammadex, and these are all impurities of the Sugammadex related compounds. Sugammadex has more than 20 reported, maybe even 30 reported impurities, depending on how you make the compound. The random derivatives, as I said, the cyclodextrins that you typically use as excipients. What are the advantages? Usually it's a one-step synthesis, aqueous-based, so it's technically green, cheap to produce, and usually they are already available, so you can just buy them from the supplier. But it's a mixture. So you may see batch-to-batch reproducibility issues. You may see issues if you change suppliers because the fine patterns or the composition may not be necessarily the same as with the other one, depending on how you make it. Then the more challenging part, it comes with the analytics. So you inject something as an active ingredient that has hundreds of components. So if you see a side effect, how do you tell what is responsible? How you identify degradation, how you track metabolites, how you doing PK/PD studies, how do you control the mechanism of action? So there comes a lot of challenges with this type of concept, and some of them may be okay for certain diseases, but certainly there are a lot of question marks around that. So just to compare from a paper, that the two hydroxypropyl-β-cyclodextrins that were used for Niemann-Pick C, as you can see based on their MS spectra, they're not nearly the same. The main components of the VTES compound are basically not even present in the other one. So they are not the same material which you use for the same disease. Funny part, both are compliant to the pharmacopoeia. So they are both regulatory wise approved. They're just, but as you can see, they are different. So as a as a punchline or a closing thoughts, single isomers, if you know the purpose and the target compound, they are definitely your choice. But they will be more difficult to make and more expensive to make. Uh, and of course, here it's a new chemical entity, unlike the established excipients, which have an established safety profile and a lot of available data with humans. Here you can face uh, what surprises, like with the Sugammadex. It may occur that, okay, it's causing an anaphylactic reaction and in a certain percentage of the cases. But on the other hand, you had a very good understanding of what happens with the compounds during the production and in the human body. Of course, coming back to the cholesterol question, if we don't know the target, then how do we develop such kind of compounds? So for the cholesterol type of application, this seems to be currently not a suitable route. Random derivatives, they're repurposed, cheap, green, commercially available with known safety profiles. But if you want to really understand what's happening with them in the vial or in the body, you will probably have to face a lot of limitations and make a lot of compromises. And here comes the question. So these are approved excipients. They're supposed to be inert. Even if we consider them functional excipients, they're supposed to be inert. So can we say that up to this dose, it's an excipient, and if you go above, it becomes a therapeutic agent because we realized later that it can have an effect on the human body? And will the specifications be the same for the excipient and the active ingredient? The two I showed you, two active ingredients, both could be approved as pharmaceutical as excipients, and as active ingredients, the specification is not as strict. So from a regulatory perspective, this will lead to a lot of challenges. So with that, I'd like to thank you for your attention, just as to confront you with the hard reality. So we have seen here a lot of wonderful research and fantastic companies who are developing cyclodextrin-based products since Sugammadex, so 2008. No new cyclodextrins were introduced to the pharmaceutical market, which is a long time. Uh, and how I see that this new application to be them for them to be used as active ingredients pose two big challenges. We have to understand, and I'm not talking about the scavengers, but the cholesterol influencers. So we have to understand how they work first, because without that we'll just shoot in the dark with inefficient compounds. And after we understood how they work, we have to design cyclodextrins that are not where we don't need 2 grams per body weight kilogram. So if you have to inject 50 grams per treatment to a patient, it's not a drug, uh, at least in my view. And for that, probably a lot of collaborations will be necessary. So what I would like you to remember are the dilemmas if you ever consider developing cyclodextrin-based active. What is an excipient? What is an active ingredient? How is my cyclodextrin working? Is it a scavenger or is it a cholesterol modulator? Should I use a single isomer or can I go with a random compound? Uh, and would I do I want to go with a new chemical entity or I repurpose something, because of course hundreds of cyclodextrins are already readily available from the market. And with that, I go and take a rest and hope to have many of your questions in the chat box. So I'll stop the screen share and please shoot any questions that you may have or anything you're interested in, ideally in the field of cyclodextrins as active ingredients. But of course, I'm really happy to answer other questions as well. Okay, I'm very happy if okay, it was inspiring and you got some new ideas. Is there a way to look back on this PPT or the recording? So, yes, the PPT itself we're not sharing, but everyone who is registered, which I suppose is all of you, we receive a link to the recording. So all the master classes are uploaded to CarboHyde's YouTube channel. So not just for you, anyone in your company or in your network who could be interested. It's a free channel, and anyone can rewatch the recordings for unlimited time on YouTube. But you'll receive the link in 1 or 2 days. What is the general route of administration? Oh yeah, Ricky, that's an excellent question, and indeed, I forgot to answer that. So, okay, it depends on the treatment, but usually, unless the therapy validates other type of use, it's parenteral. So as I mentioned, cyclodextrins are unable to cross biological membranes, which is also true to the GI tract. So the bioavailability of cyclodextrins are in the oral bioavailability, I mean, are in the range of 1 to 5% depending on compound. There's just the methylated cyclodextrin that's 12, and that's the top. So accordingly, if you treat something that's systemic or a chronic disease, the administration route is always parenteral, usually intravenous. So all the cancers, the Alzheimer's, this Niemann-Pick C, they all came parenterally. Of course, the wound healing is topical. Or when I talked about the COPD or the asthma, that was an inhalation treatment. Or uh, or the alpha cyclodextrin for weight loss, that's an oral medication because we don't need them to the system. Or the lipofuscin accumulation in the eye. I am not sure if it was an eye drop or an intravitreal injection, but certainly something local to the eye. 90% of the cases, it's parenteral administration because of the lack of membrane crossing. We also have a topic in-house at CarboHyde. How could we develop cyclodextrins that would be orally bioavailable? That would be wonderful, but currently unresolved problem. Okay, thank you for the compliments. I really many different types of Charbel asking many different types of oxidized cholesterols. Does cyclodextrin modified cyclodextrin target all of them or only one subtype? So that's a good question, and maybe I should link you to Cyclarity people because I don't know the answer for that one. So what I do know is that the target was the 7 ketocholesterol. So that was the bad guy that they claimed uh, and they wanted to remove. And for that, they tested the specificity for the removal, but I don't recall data if and they compared it to unmodified cholesterol, but I don't recall any data if they checked if it also removes other type of cholesterols, other type of oxidized cholesterol. So no idea about that, but it's a very good question. I hope that something they will test in clinics, not just the 7 ketocholesterol. So just partial answer. Sorry about that. Why do you think CDs are not seen in biologic therapeutics for parenteral administration frequently? Well, I think that will change in the future, but certainly you're true. It's not a very uh, typical for cyclodextrin to go in that space. So we know of a lot of companies who take seriously the aggregation prevention effect of cyclodextrins in monoclonal antibodies. Mainly Genentech and Roche works a lot on those applications. So to replace or combine polysorbates with cyclodextrins to have a better stabilizing effect. So on the protein front, I can feel that coming, but it takes a lot of time. So I don't even know about any compound which is in clinics with the concept. We know that there are quite a few vaccines, okay, quite a few, maybe an exaggeration, but there are some vaccines on the market that contain cyclodextrins for different purposes. So stabilization, one. Uh, Cryoprotective effect of cyclodextrins is another one, and cyclodextrins are also known, some cyclodextrins, to have an adjuvant type of effect. So there are some vaccines like J&J's COVID vaccine or Daiichi Sankyo's one intranasal influenza vaccine that contains cyclodextrins as adjuvants. So in that space, the vaccine development, they have the potential, and not just the potential, they're on the market, but rare. Why? I don't know. For the protein stabilization, I do know that it could be probably cost. You need a fairly big amount of cyclodextrin to stabilize the proteins compared to the 0.1% typical polysorbate level. And cyclodextrin is more expensive than polysorbate, so the cost factor would not really justify the use of cyclodextrins if other solutions work also well. For the vaccines, I think it's not that widely known what kind of potential cyclodextrins may have in that space. Uh, so I think I truly believe that in most areas it's a matter of time. And for gene therapy, all the data are still preclinical, not even in vivo. There are some cyclodextrins that can be used to improve the transfection of oligonucleotides. We also have a program like that, but it's 10 years at least until it gets to something close to the market. So Henry couldn't attend the beginning, and he's wondering about CD therapeutic effect when administered via intra-articular injection. So, something I haven't seen at all. Uh, so I don't really have a firm opinion on that. Neither have I seen any company who did intra-articular administration with the cyclodextrin. So intravenous, intramuscular, yeah, even subcutaneous now. Uh, Another type of like this intrathecal, but intra-articular, what would be the purpose of that administration? I don't really have a good answer for that one. I'm sorry, Henry. But if you have a particular why you would use it like that, maybe we could figure it out. I don't know. Well, okay, I didn't say much, but okay, you're welcome. Okay, guys, our time is up. Okay, still Lance questions. Len is familiar with Cyclarity. Yeah, they're based in Novato. Their city derivatives that's designed only to target 7-ketocholesteryl. Yes, that was the intention. I agree. I also have the same information. The question is it still selective? So I seen proof that it's selective for 7-ketocholesteryl against cholesterol, but I haven't seen data how other types of smaller cholesterol analogs would behave with this one. So let me just tell you very quickly the story and then we conclude. So for Sugammadex, the idea was to design for rocuronium, and that was very specific and selective. But it was not only rocuronium that Sugammadex was binding, so it does have not this super huge affinity, but it does have affinity towards other intrinsic compounds. And rocuronium has a steroidal structure, so steroidal compounds like cholesterol or other similar compounds, dominantly hormones, were encapsulated by Sugammadex, not removed, but encapsulated, and their PK was influenced. So, okay, it's not necessarily. So one of the suggestions on the leaflet of Sugammadex is that the anti-contraceptives that you take when after the use of Sugammadex probably will not work in the next 2 or 3 days because Sugammadex also messes up uh, their receptor binding and stuff like that. So something similar may also happen in the case of Cyclarity's program, but Not with the cholesterol, but okay, something else, something that's similar, something that is still binding to the cyclodextrin. It's a completely feasible idea. So, time is up. If you have any further questions, I'm really happy to answer them offline. So you know our contacts. And thank you all for joining. I really enjoyed this. Hope that I could share some new insights. You enjoy your day, evening, and see you at the next masterclass, hopefully. Thank you all, guys. Thank you. Take care. Bye bye.